This presentation from the 3M Visual Attention Service will focus on the human visual system. In this presentation, I'll be covering limitations of the human visual system, what happens when we first view a scene, some background on visual attention modeling, which is the basis of the 3M Visual Attention Service. We'll do a couple of experiments, one on shape and color, the other on visual processing, and then finish up with how the 3M Visual Attention Service works. Human visual system limitations. Turns out when we first view a scene, we don't notice or process the majority of visual stimuli or visual information. Our eyes are actually producing more data than our brains can process in a reasonable amount of time. Another limitation is that our eyes don't provide full, high-resolution, high-color information across the entire field of view. Only the center 2 degrees of visual angle, where the light from the image is falling on the fovea, do we get high-resolution, full-color information. The fovea has a high concentration of cone photoreceptors, good for both color and resolution. Away from the fovea, there are more rod photoreceptors. These provide low-resolution black and white information. Rod photoreceptors are actually quite good in low-light conditions. Here's a drawing of the human eye. The front of the eye you have the lens. The lens images the scene onto the back of your eye, or the retina. In the center of your retina, there's the fovea. This is where you have the high concentration of cone photoreceptors, good for color and resolution. As you move away from the fovea, there's an increasing concentration of rod photoreceptors, which are good for relatively low-resolution black and white imaging. You can also see the optic nerve and where the optic nerve exits the eye. The relative acuity or resolution of the human eye is highest right in the center of the fovea, where you have the high concentration of cone receptors. As you move away from the fovea and have the increasing concentration of rod receptors, you can see that the relative acuity or resolution drops off. You can also notice the blind spot. That's where the optic nerve exits the back of the eye, and there are actually no photoreceptors. The main takeaway from this slide is that resolution drops as you move away from the fovea. Here's an example to show in greater detail how the human visual system interacts with a scene. On the left-hand side, we've got an image of a real-life scene. On the right-hand side, we have a simulation of what the human eye would see at a moment in time. In the center, or the fovea, the center 2 degrees of your field of view, you get full resolution, full color detail. As you move away from the fovea, the color accuracy falls off, as does the resolution. So what happens when we view a scene? When we first see an image, there's rapid movement of our eyes as we scan the entire field of view between points of fixation. A fixation is a spot where we maintain our visual gaze on a single location. What we're doing during this rapid movement period is we're sampling the scene with our high-resolution fovea. Over time, using this information, our brain stitches together a high-resolution representation of the scene. The initial fixations, or points where we will maintain our gaze, are well predicted by certain imaging properties, things like color, shapes, contrast, edges, and faces. Visual attention models. For years, industry and academic research has been taking place in vision science and computer vision. Out of this work have come predictive computational models. These models predict where people will initially look when they view scenes. The models are based on the findings from behavior research and vision science, or based upon imaging statistics. As I mentioned before, when we first view a scene, we don't notice or process the majority of visual stimuli. When we first see a scene, our brain decides what we should pay attention to. These initial fixations are well predicted by certain imaging properties. Alright, we're going to move on to our first vision science experiment that I hope will help to convince you that we don't notice or process the majority of visual stimuli. For this experiment, I'm going to cycle between two images, and I'm going to ask you to look very carefully to see if you notice any changes. Here we go. Did you notice any changes? Let's look at the two images side by side. If you look carefully at the image on the right, you'll see that the aircraft has a jet engine attached to the wing. Unfortunately for the people in the left image, they're getting onto a plane that's missing the engine. I'll run the sequence again. Pretty easy to spot now that you know the difference. So how does VAS work? VAS algorithms process important visual cues, things like shapes, colors, edges, contrast, and faces. These are things that are likely to get your attention when you first view a scene. Now we'll move on to the second vision science experiment. This one is all about shape and color. I'm going to again sequence through a few slides. In this case, I'm going to ask you to see how long it takes you to find the blue O. Here's the first slide. Pretty easy here. Also pretty easy with this slide. Now it gets a little more complicated. Have you been able to find both blue O's? Okay, so what's going on? Turns out we have a portion of our brain that is responsible for evaluating shapes. If you have a unique shape in your field of view, your brain can recognize it very quickly. We also have another part of our brain that is responsible for evaluating color. If you have a unique color in the field of view, it's very easy to pick up. Things get a lot more complicated when you have a mix of shape and colors. In this case, two parts of your brain have to work together to figure out what's going on, and it takes a little time. Color and shape are two of the factors that VAS algorithms evaluate. There are also many other factors. Things very quickly get complicated as you add factors. How VAS works. VAS algorithms process important visual cues, things like colors, edges, faces, shapes, and contrast. These are the things that are likely to get our attention when we first view a scene. The decision-making process that our brain goes through when we decide what to pay attention to is part of everyone's pre-wired neural architecture. Test results have shown that VAS is very consistent across gender, age, and culture. These are important biases for marketers to consider, but they don't come into the picture until after we've decided what to pay attention to. VAS accuracy has been confirmed with eye tracking studies. The 3M Visual Attention Service is a web-based scanning tool that you can use to enhance the visual impact of your designs. VAS predicts which elements of content viewers will see when they first look at a scene. This allows you to apply science to the design process. VAS is fast, convenient, and affordable. You can use VAS to ensure that your creative gets the audience that it deserves. Thanks for your time.